I went over the basics of single-cell ATAC-seq, showed you how to find and download data and which files you need. We did pre-processing, combined multiple samples, dimension reduction, clustering and plotting. We estimated gene activity. We did differential accessibility between groups of cells, and we plotted genome coverage, along with some simple comparison. Alright, so just a brief introduction into ATAC-seq. Basically, the main idea is that we're going to sequence the accessible regions, not the ones that are wrapped tightly. So these transposases, they can bind to the accessible regions, but not these regions. They chop up and tag the pieces that they could reach. So we're left with a bunch of fragments that we can then map to a reference genome. In single-cell ATAC-seq, we do this in each individual nucleus. So instead of genes, we have peaks. And how we come up with our set of peaks or features, we take all of the data and map it to the genome, and then we do a peak calling across all cells in that sample. So at every significant peak, we'll label that as a feature. So in each sample, you're going to have a slightly different set of features, especially if they come from different cell types. So after we've identified the features from each sample, we then create a counts by read matrix for every individual cell, where we have the cells and all these regions. We have the chromosome it mapped to and the position on the chromosome. And the big distinction here from single cell is instead of 20 to 30,000 features or genes, we have anywhere upwards of 200,000 features in a single cell ATAC-seq data set. For this tutorial, I'm going to use this recently published ATAC-seq data from this Nature Communications paper, just because I thought it was an interesting and simple example. They just took HSCs from young mice and old mice and then did single cell ATAC-seq on them and some other stuff, but I'm just going to focus on the ATAC-seq. You follow the geo-accession number. We're just going to download the raw tar file, which should have the counts that have already been processed through the 10x software. So you can just go ahead and download this tar. So they had a bunch of files in that tar. You can just go ahead and unzip that. But the only ones we really need are the H5, so the peak by cell matrix, the fragments file, the fragments index file that ends in .tbi, but I'll show you what those files should have in them. So the fragments TSV file has the chromosome, the position, and the cell barcode. That single cell CSV file has the barcode and then some metadata, and we'll need these later on when we do the pre-processing. So I'm going to be using Signac, and I'm going to be doing more or less what they do in their online tutorial, which is pretty thorough. But I'll explain a few things that I think are important and also change a few things. All right, so we're just going to start in a new RStudio notebook. The first thing you're going to want to do is install a few packages, HDF5R, and then of course we need to install Signac and Seurat if you don't already have them. Then we can go ahead and load those in. And then we're going to start by reading in the H5 files. I'm just going to start with one sample, and we're just going to read it in as counts. And I'll make this notebook available on my GitHub so that you can just copy and paste it. So if we look at counts, we just have this 187,000 feature by 5,028 cell matrix. As you can see, we have as features the mapped coordinates of each peak. Let's just go ahead and read in that metadata, which is just a data frame. And then we'll need to read in the fragments file. So there's an important note here is that the TBI file needs to be in the same directory as your fragments file. And if it's in your working directory, you'll need to put a / or you'll get an error because it won't know what directory to look in for the index file. The TBI file might end with a GZ. And if it does end with a GZ, you need to gunzip it or you'll also get an error. So we'll read that in with this create chromatin assay. And we're passing counts and then the separators of the features. So again, if we looked at counts, each feature is separated by a colon and a dash. If your data have different separators, you need to change that. And we also need to specify the genome. If it was human, I would put HG19. But anyways, let's read that in. And now we can create our Seurat object with the chromatin assay we just made. We're setting it as peaks. And then we're also combining it with the metadata frame that we made earlier. And don't worry about this. So if we look at our data now, we have our Seurat object with 5,000 cells, and we filtered out some of those features. And then we can look at the metadata. You see we have the cells and some of these different metadata that the 10x pipeline calculated and that we'll use to do filtering. But first, we'll have to install a few more packages. We need this ensemble database for a mouse. If you're using human data, you need this database. And we also need genome InfoDB and BioViz base. But we'll go ahead and load these in. We'll load in those annotations. So we're going to get the range of different genes from the mouse database. If we look at the annotations database, it's just the ensemble IDs, where that gene is, and its gene symbol. So we're going to add this into our data, this annotation function. So we need to do this before pre-processing because now we can quantify where the peaks actually fall relative to where genes are, and specifically gene promoters. First, let's calculate one more metric, this nucleosome signal, which calculates a ratio of the fragments that are between 147 and 294 base pairs and those that are less than 147, because the average length to wrap around one histone is 147 base pairs. And that way we can calculate the ratio of fragments from mononucleosomes versus fragments that are nucleosome free. And this will just add another column in the metadata. And then we'll add another column using our annotations so we can get the transcription start sites based on the gene locations to calculate the TSS enrichment. This will take a minute or two. Since that's done, let's look at the metadata now. And you can see we added the nucleosome signal and also TS enrichment data. So next, we're going to calculate the blacklist region ratio. So we have a column called blacklist region fragments. So the number of fragments that map to blacklist regions, which are kind of just junk regions that have been annotated. They have high signal in different NGS experiments independent of cell type or experiment. So they're regions we know are no good. So we're just going to add this new column, blacklist ratio, which is the blacklist region fragments divided by the total peak region fragments. And we just have this new column. And then we're going to calculate the percent reads in the peak, which is just this column we already have, divided by this column that we already have, and then multiplied by 100. And just know that these columns can sometimes be named slightly different, like past filters can sometimes be ATAC fragments. So now we have all the QC metrics that we need. Let's go ahead and just look at those on some violin plots. Basically, we're just going to filter outliers here. So we're going to keep the majority of these. So get rid of the outliers. We're going to get rid of the cells with a low number of reads that actually map to peaks. Just going to be keeping these. And then we're going to get rid of anything with the high blacklist ratio, keeping these. We're going to get rid of things with the high nucleosome signal. And we're going to get rid of things with a low TS enrichment and keep everything above a certain threshold. So we could just pick our thresholds based on looking at these violin plots, but I like to be a little more objective. So instead of this, I'm going to set several thresholds using the quantile function. So I can find the threshold that differentiates between bottom 2% and everything else, and the peak region fragments, and then likewise for the 98th percentile, and then the same thing in percent reads, blacklist ratio, nucleosome signal, and TS enrichment. So if I run those, let me just print those out to show you. So you can see here, it picked these numbers for us. And then we can use that subset function, but plug in these variables instead of numbers and use that to subset our data. So this is what's actually filtering out all the outliers here. So we're going to be left with a clean Seurat object. So we filtered out a few hundred cells, but we still have 4,500. So now the pre-processing is done and we can do normalization and dimension reduction. So first we'll normalize the data using this run tf-idf. And then Seurat makes us set the top features, but we're actually going to set the cutoff as Q0. And as you can see, the features equals the variable features. We're going to do run SVD, which is analogous to PCA in typical single cell analysis. I'm just going to plot the correlations of these top 10 components we just calculated, just to show you that we have to get rid of the first component. So we're not going to use that in any downstream analysis because it's highly correlated with that. So it's basically an artifact that we want to get rid of. So let's do the UMAP and neighbors and clustering. This is going to be a lot like single cell with a couple distinctions. We have to pass reduction LSI, and then we have to only use 2 to 30 dimensions, again, because we don't want to use that first dimension. And then they recommend that you use algorithm 3, and then we're just going to go ahead and plot that. And so we have our UMAP, and of course the dataset I chose was only HSCs, so we get one primary grouping of cells with a few different clusters within it labeled. But to show some differential accessibility, I'm going to import both samples, and to do that I like to make a function just so I don't have to write everything out twice. So inside this function, I basically just include everything that we just did, and we're just going to pass a path to those 3 files for each of the samples, and then everything else here is going to be exactly the same. In this example, just for simplicity, I'm not going to be integrating. I'm just going to go ahead and pass those 6 file names and load in young and old. Oops, I actually have to run the previous block of code. All right, once we have both of our samples in, I'm just going to add a new metadata column to differentiate between the 2 of them. And then I'm going to merge them. So this is going to take like 30 minutes, unfortunately. It's just a symptom of the data being really large and R being slow, but just let it run. It'll finish eventually. But if we look at data now, we have 7,800 cells and 177,000 features. So we stopped at the pre-processing earlier and we didn't run any of this. Now we have to do that on the merged object. So we'll just basically do the same thing that we did to the individual sample earlier. Once that's done, we'll run the UMAP and find the neighbors, again, skipping that first dimension. And then we're going to run find clusters. And I like to just start off with a resolution of 0.5, but we can change that depending on what the UMAP looks like. So we can plot the UMAP based on the latent clusters. And if we want to look at the UMAP labeled by the dataset column that we added, which just differentiated between the young and the old, you see that the data are separated into these 2 different clusters. It's pretty similar to what they had in the paper as well. I think they do an integration. They had a little more overlap. And I'm just going to go through some simple data analysis now. So the first thing we're going to do is add gene activity, which is a function that assumes the gene activity based on the chromatin accessibility near that gene. All right, so that takes a couple minutes. But after that's done, we have a matrix that's a gene activity by cell matrix. So the estimated expression of RNA based on the ATAC data. So we're just going to save gene activities as an assay object to the RNA slot of data. And then we're going to normalize that. And then we can look at that RNA slot we just saved. Again, we have these 21,800 genes and our cells and our gene symbols. So the default assay is still our ATAC data, but we can set the default assay to RNA if we want to do plotting and other functions. And then we can do typical single-cell RNA-seq stuff like feature plots and then plotting the expression of these genes. And I've just picked a couple HSC markers for demonstration. And you see we get what we're used to when we're doing single-cell RNA-seq. So this is a useful method if you're trying to call clusters for cell types, etc. But of course, we only have one cell type here. Anyway, I'm going to set the default assay back to peaks, and then I'm going to do differential accessibility between our old and young cells. So again, if we look at the data, metadata, I had earlier added this dataset column, which is just young or old. So I'm going to subset the cells by that, where you filter that metadata based on your variable, and you pass that to row names to get the cell names. So we're going to use LR, and we're going to use the peak region fragments for the latent variable. So this will give us the differentially accessible peaks between the old and young cells. All right, so now we have DA peaks, or differentially accessible peaks, which is just a data frame of our features that were significant between our two testing groups. But all we have right now is the feature, so we don't really know where this is. Of course, we can plot it, but let's go ahead and add the closest gene to each one of these regions. So if we run this closest feature and we pass data and we only want the regions from DA peaks, see, all this does is return a data frame that's the same length as our DA peaks with the gene name and some of these other symbol IDs. But all we really care about right now is the gene name. But if you really cared, it also tells you the distance from the gene as well, which could be useful. But we can just call the gene name column, and we're just going to make a new column in DA peaks called closest gene. And actually, I'll just add the distance as well. So after running that, now our DA peaks has the closest gene and the distance. But anyway, here are your differentially accessible peaks. You can filter by up or down, etc. But let's go ahead and plot one of these. I'll just pick the first one and use the coverage plot function. So we're just getting the first row name in DA peaks, which is the region. And I'm going to plot 10,000 upstream and 5,000 downstream and group by dataset, which again is old or young. So you can see this is in a non-coding region because we don't see any gene below down here, but we have this large peak in old and not in young. We could try another one, the second most significant one. And here, these peaks are in this promoter region or overlapping with these genes here. And then we could also plot some violin plots and feature plots based on a region of interest. Let's just do this one again. Here we're just plotting a violin plot, picking that same feature or region and grouping by young or old. And then we're just going to show the accessibility on a feature plot as well. So here was a pretty basic introduction into single-cell ATAC-seq analysis. I went over the basics of single-cell ATAC-seq, showed you how to find and download data and which files you need. We did pre-processing, combined multiple samples, dimension reduction, clustering and plotting. We estimated gene activity. We did differential accessibility between groups of cells, and we plotted genome coverage, along with some simple comparison